Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. You're going to hear my voice but not see me because I'm zoomed in on these two models. Today what we're going to be covering is the ventricles of the brain, which are some fluid-filled chambers, and the meninges, the coverings of the brain. Now if you're following along on the list of things to know, first and foremost, if you're watching these videos, you know that they're intended for use by students who are enrolled in my anatomy and physiology class, at Del Mar College. If you're not in my class, your instructor may use different models, they may use dissections, they may use different structures, different names. So learn the material the way that your instructor wants you to learn it. If you find my videos helpful, please hit like, please hit subscribe if you want to subscribe, but at least hit like. Now, if you're following along for my class, we're going to be on the list of things to know. We're going to be at the bottom of page 32 and the top of page 33. I'm actually going to start on page 33 and then work backwards. Okay, we're going to cover the ventricles on page 33, then we're going to cover the meninges on page 32. So if I look at the outside of the brain, it doesn't appear to have much empty space inside of it, but it does. And I did some drawings of this and of the meninges. Hopefully you've watched that video first, and then you can come back and see this and see how I'm going to test you on the models. Those empty chambers, if I could break the top of the brain open, there would be some empty chambers on either side of the corpus callosum, and they're filled in with this plastic fluid. If I could dump water in there, somewhere in the thalamus, there's two little holes that come together and form another ventricle, and there's another hole that runs down and forms a fourth ventricle. And so they're all buried inside the brain. If I open the brain up and I start to take it apart, let me pull this model aside for a second, push that out of the way. I can see here on either side of the corpus callosum, this is front, this is back, these two big plastic pieces here would be some fluid. If I poured fluid in there, stuck this in a freezer and froze it, I would have ice cubes in the shape of the container, so to speak. So that represents where the space was, the chamber called a lateral ventricle. There's right lateral ventricle and a left lateral ventricle, or you can just say lateral ventricles. Now, I'm going to pop off the corpus callosum. There would be a septum pellucidum here. We don't see it on this model. And then on either side of the fornix here, there's a little hole, and we can't see it, but there's a little hole that drains into the thalamus. So if I poured fluid in those empty spaces, it would drain right into the middle of the thalamus. So if I continue to take this model apart, what I get down to is the brain stem. And I can see how the lateral ventricles run here and here straight back to these points, but then they run underneath on either side. That's all lateral ventricle here. There would be a septum pellucidum there. And then I have another lateral ventricle that runs underneath here. It's kind of hard to see when I'm manipulating it, but if I pop this part of the diencephalon off, I can see how this lateral ventricle wraps around where the thalamus is and underneath. There's another one on the other side. They're going to drain right about here through to a little hole inside the thalamus. So if I pop this part off, I can see how it sits three-dimensionally. Here's the lateral ventricle. This little connector right here is called the interventricular foramen. People look for a hole, but the whole thing is a space. So that's your interventricular foramen. And then this little structure down here would represent the third ventricle, sitting inside the thalamus and the hypothalamus. We have the cerebral aqueduct, and we have the fourth ventricle here. Okay, so if I pop this off, you can see how it would fit inside there and sit around this structure. So if I look here, lateral ventricle, interventricular foramen, the two little connectors there. Here's the third ventricle, kind of looks like a little moose head with an eyeball. And then that's the the cerebral aqueduct and the fourth ventricle here. Now, the fluid that is filtered out of our blood and into these chambers is filtered by a series of specialized capillaries called the choroid plexus. That's why there's red on here. We're not going to worry about that on this model. Now, we also have this other model that has the ventricles on a stand. So this is still your lateral ventricle. If I twist it around, I can see another lateral ventricle here. I can see, if I look at it this way, those two little connectors, you see how it looks like the letter Y, and then these are sitting on top? Those two little connectors there and there would be the interventricular foramen. It's not much, but it's that little connection. This would be the third ventricle here. It looks like a little moose's head. Let me rotate it this way. So lateral ventricle, interventricular foramen, third ventricle. The fluid would flow down here through the cerebral aqueduct into the fourth ventricle. On this model, if it's painted pink, and all four ventricles, their pink stuff represents what we call the choroid plexus, the vessels that filter out the cerebrospinal fluid. And that cerebrospinal fluid would flow up here through the interventricular foramen to the third ventricle, where the thalamus and hypothalamus are, through the cerebral aqueduct, down here to the fourth ventricle. I hope you can see how that fits together. Okay, so this is one of the more difficult things to get across to students when you're teaching brain anatomy. Now, I'm moving out the way and I'm going to bring in two other models, and these two models are going to show us what we call the meninges. I call this one the Meninges model. We call this one the handle head model because it looks like his head has a little handle. They're going to show us a lot of the same structures. Now, if you watch the videos that I talked about earlier, where I drew out the meninges and I drew out the ventricles, the meninges are pretty tightly wrapped around the brain. And those meninges have some gaps between them. We have one called the dura mater, the outermost layers. Then there's the arachnoid mater. Separating those is a space called the subdural space. Underneath the arachnoid mater is one that's right up against the surface of the brain called the pia mater. And separating those is a space called the subarachnoid space. So if I could peel all them off, or actually if I could show you this model, let me move this one for a second. If I look at it from the front, this side of the model looks nice and clear and clean. This side of the model is very vascular with this white film covering it. That white film or white membrane represents the arachnoid mater. So if I ask you for this white covering, that's the arachnoid mater. They've peeled it off the other side. These little white dots right here are called arachnoid granulations, and they're like little pieces that stick up. If you were looking at the tips of my fingers and I twisted them, then we're seeing the tips of the fingers by looking down at this, but they would be holding up this piece right here, which would be called the dura mater. Now, I'm going to grab a sheet of paper to illustrate something, and I'm going to show you on a different model. If I took this brain on the half shell, okay, and I take this piece of paper and fold it, I could slide it all the way down into where it hits the corpus callosum. And then I can wrap the paper out around this. I wrote some notes on there. And I could completely cover the brain. That covering would be called the dura mater. Now they've taken some scissors and they cut most of the dura mater off and peeled it off so we can see the arachnoid mater and the arachnoid granulations. And then it leaves us this little fold right here that fits inside the cerebral hemispheres like that. Let me show you this way. Okay. So this little flap on either side here represents a piece of the dura mater. At the top edge of the dura mater, there's a big blue blood vessel running through here that's called the superior sagittal sinus. It's a long blood vessel that runs all the way through here. It collects a lot of blood, and the arachnoid granulations collect cerebrospinal fluid and drain it back into that vein. Now watch. If I pull the two pieces of dura mater up, you see this flat piece right here that fits in between the hemispheres? That's called the falx cerebri, F-A-L-X, pronounced falx, and cerebri, meaning cerebrum. It is a fold, falx means fold, of dura mater that fits between the cerebral hemispheres. It actually has arachnoid and pia mater in it too. So these two pieces here would be your dura mater. Of course, they cut most of it and removed it. Falx cerebri. This blood vessel on top is called the superior sagittal sinus. We saw it from this view, but there we see it again. This is called the inferior sagittal sinus, and that would be falx cerebri. Okay? So on this model, I can see dura mater, superior sagittal sinus, arachnoid mater, and the arachnoid granulations. The clear membrane that would be covering this, like if I put saran wrap over it, would be pia mater. If I pull this up, I see the falx cerebri and the superior sagittal sinus there. Now the reason we have the handle head model is because it represents the exact same stuff, only missing the brain, and we have the bone tissue here. You see the coronal and sagittal suture of the skull. So this structure here, that flat spot, is this one. That's your falx cerebri. Only this one has a piece, this one has the bone on top. You can see some spongy bone and some bone marrow there. Now, if I lift it out and tilt it, you can see the superior sagittal sinus and the falx cerebri. The little flaps that would have been up against the skull would be dura mater here. Also, if I look inside the skull, you can see the dura mater is this white lining. It's almost like the lining of a helmet. Okay, it lines all the inside of your skull. Now, if I do this, and it's a little bit tricky to see, but when I have the dura mater going over the cerebrum, the dura mater actually tucks into the folds of the cerebral hemispheres, and it tucks into any other nook or cranny or crevice that it can fit. So when I put this other brain back together for a moment, give me just a second, I wasn't prepared. I should probably reshoot the video, but I'm not going to at this point. You guys can follow along for a second. So when I'm looking at the brain like this, the cerebellum is not actually attached to the cerebrum. The cerebellum is attached to the brainstem, and they can be separated. And I can actually fit a fold of dura mater inside of there. And because of the shape of the cerebellum, now let me move that, because it has this little point on top, it makes a little tent over the cerebellum. So these two little flaps represent a piece of that meningeal structure. This would be our falx cerebri. It comes down and flattens out and forms these two little wings that are called the tentorium cerebelli, or cerebelli. That's the tentorium cerebelli, like a little tent over the top of the cerebellum. Now, if I take this model where I see the dura mater inside, and I stick the cerebellum in there, I can see how the tentorium cerebelli separates the cerebellum from the cerebral hemispheres. It would all be in there something like that. Okay? So, on this model, I can see dura mater and dura mater. I see the falx cerebri and superior sagittal sinus, and I see the tentorium cerebelli. Finally, if I look at the cerebellum, underneath it kind of dips in and comes back out, and it creates a little nook there, spot, where the dura mater can fold up. This little fold right here, this would be your superior sagittal sinus. This is where the tentorium cerebelli was attached, but we cut it with a scalpel and removed it. This little fold that goes down to the spinal cord would fit right inside that little groove of the cerebellum, and that is called the falx cerebelli. Falx cerebelli. So, one last run through. I'm going to go through both models for you without me moving a lot of stuff or making you wait. These two flaps are dura mater. The dark line is the superior sagittal sinus. The clear covering on this side would be pia mater. The white covering on this side would be arachnoid mater. And these little white bumps are called, or dots, are called arachnoid granulations. If I pull the dura mater up, this flat piece here is called the falx cerebri. The big blue blood vessel would be the superior sagittal sinus. If I take this model, I still have underneath here dura mater. I have the falx cerebri and superior sagittal sinus. I have the two flaps of the tentorium cerebelli. And then in here I have the falx cerebelli. And this is all dura mater. All right. Look, I hope you learned something. I hope you had as much fun as I did. Pick 3 or 4 structures, repeat them in the same order till you can't stand it. Learn the ventricles, learn the meninges. We'll talk about them in lecture. Again, hope you had as much fun as I did. I'll see you on the next video.